Plant microbiome. The plant microbiome, also known as the phytomicrobiome, plays roles in plant health and productivity and has received significant attention in recent years. The microbiome has been defined as a characteristic microbial community occupying a reasonably well-defined habitat which has distinct physio-chemical properties. The term thus not only refers to the microorganisms involved, but also encompasses their theater of activity. Plants live in association with diverse microbial consortia. These microbes, referred to as the plant's microbiota, live both inside the endosphere and outside the episphere of plant tissues, and play important roles in the ecology and physiology of plants. The core plant microbiome is thought to comprise keystone, microbial taxa that are important for plant fitness and established through evolutionary mechanisms of selection and enrichment of microbial taxa containing essential. Plant microbiomes are shaped by both factors related to the plant itself, such as genotype, organ, species, and health status. The health status of a plant has been reported in some studies to be reflected by or linked to its microbiome. Overview. The study of the association of plants with microorganisms precedes that of the animal and human microbiomes, notably the roles of microbes in nitrogen and phosphorus uptake. The most notable examples are plant root arbuscular mycorrhizal and legume rhizobial symbioses, both of which greatly influence the ability of roots to uptake various nutrients from the soil. Some of these microbes cannot survive in the absence of the plant host. Obligate symbionts include viruses and some bacteria and fungi, which provides space, oxygen, proteins, and carbohydrates to the microorganisms. The association of fungi with plants has been known since 1842, and over 80% of land plants are found associated with them. It is thought fungi helped in the domestication of plants. Traditionally, plant-microbe interaction studies have been confined to culturable microbes. The numerous microbes that couldn't be cultured have remained uninvestigated, so knowledge of their roles is largely unknown. The possibilities of unraveling the types and outcomes of these plant-microbe interactions has generated considerable interest among ecologists, evolutionary biologists, plant biologists, and agronomists. Recent developments in multiomics and the establishment of large collections of microorganisms have dramatically increased knowledge of the plant microbiome composition and diversity. The sequencing of marker genes of entire microbial communities, referred to as metagenomics, sheds light on the phylogenetic diversity of the microbiomes of plants. It also adds to the knowledge of the major biotic and abiotic factors responsible for shaping plant microbiome community assemblages. The composition of microbial communities associated with different plant species is correlated with the phylogenetic distance between the plant species. That is, closely related plant species tend to have more alike microbial communities than distant species. The focus of plant microbiome studies has been directed at model plants, such as Arabidopsis thaliana, as well as important economic crop species, including barley Hordeum vulgare. Plant microbiota. Cyanobacteria are an example of a microorganism which widely interacts in a symbiotic manner with land plants. Cyanobacteria can enter the plant through the stomata and colonize the intercellular space, forming loops and intracellular coils. Anabina spp. Colonize the roots of wheat and cotton plants. Calothrix sp. has also been found on the root system of wheat. Monocots, such as wheat and rice, have been colonized by Nostoc spp. In 1991, Ganther and others isolated diverse heterocystis nitrogen, fixing cyanobacteria including Nostoc, Anabina, and Cylindrospermum, from plant root and soil. Assessment of wheat seedling roots revealed two types of association patterns: loose colonization of root hair by Anabina and tight colonization of the root surface within a restricted zone by Nostoc. Rhizosphere microbiome. The rhizosphere comprises the 110 mm zone of soil immediately surrounding the roots that is under the influence of the plant through its deposition of root exudates, mucilage, and dead plant cells. A diverse array of organisms specialize in living in the rhizosphere, including bacteria, fungi, oomycetes, nematodes, algae, protozoa, viruses, and archaea. Mycorrhizal fungi are abundant members of the rhizosphere community and have been found in over 200,000 plant species and are estimated to associate with over 80% of all plants. Mycorrhizae root associations play profound roles in land ecosystems by regulating nutrient and carbon cycles. Mycorrhizae are integral to plant health because they provide up to 80% of the nitrogen and phosphorus requirements. In return, the fungi obtain carbohydrates and lipids from host plants. Recent studies of arbuscular mycorrhizal fungi using sequencing technologies show greater between species and within species diversity than previously known. The most frequently studied beneficial rhizosphere organisms are Mycorrhizae, rhizobium bacteria, plant growth promoting Rhizobacteria pgpr, and biocontrol microbes. It has been projected that 1 gram of soil could contain more than 1 million distinct bacterial genomes, and over 50,000 OTU operational taxonomic units have been found within the potato rhizosphere. Among the prokaryotes in the rhizosphere, the most frequent bacteria are within the Acidobacteria, Protobacteria, Planktomycetes, Actinobacteria, Bacteroidetes, and Firmicutes. In some studies, no significant differences were reported in the microbial community composition between the bulk soil not attached to the plant root and rhizosphere soil. Certain bacterial groups, e.g. Actinobacteria, Xanthomonidaceae, are less abundant in the rhizosphere than in nearby bulk soil. Philosphere microbiome. Example: the Manuka philosphere. The flowering tea tree commonly known as Manuka is indigenous to New Zealand. Manuka honey, produced from the nectar of Manuka flowers, is known for its non-peroxide antibacterial properties. These non-peroxide antibacterial properties have been principally linked to the accumulation of the 3-carbon sugar dihydroxyacetone DHA in the nectar of the Manuka flower, which undergoes a chemical conversion to methylglyoxyl MgO in mature honey. However, the concentration of DHA in the nectar of Manuka flowers is notoriously variable, and the antimicrobial efficacy of Manuka honey consequently varies from region to region and from year to year. Despite extensive research efforts, no reliable correlation has been identified between DHA production and climatic, edaphic, or host genetic factors. Microorganisms have been studied in the Manuka rhizosphere and endosphere. Earlier studies primarily focused on fungi, and a 2016 study provided the first investigation of endophytic bacterial communities from 3 geographically and environmentally distinct Manuka populations using fingerprinting techniques and A 2020 study identified a habitat-specific and relatively abundant core microbiome in the Manuka philosphere, which was persistent across all samples. In contrast, non-core philosphere microorganisms exhibited significant variation across individual host trees and populations that was strongly driven by environmental and spatial factors. The results demonstrated the existence of a dominant and ubiquitous core microbiome in the philosphere of Manuka. Endosphere microbiome. Some microorganisms, such as endophytes, penetrate and occupy the plant internal tissues, forming the endospheric microbiome. The arbuscular mycorrhizal and other endophytic fungi are the dominant colonizers of the endosphere. Bacteria, and to some degree archaea, are important members of endosphere communities. Some of these endophytic microbes interact with their host and provide obvious benefits to plants. Unlike the rhizosphere and the rhizoplane, the endospheres harbor highly specific microbial communities. The root endophytic community can be very distinct from that of the adjacent soil community. In general, the diversity of the endophytic community is lower than the diversity of the microbial community outside the plant. The identity and diversity of the endophytic microbiome of above and below ground tissues may also differ within the plant. The seed microbiome. Individual seeds possessed high microbial diversity that was higher in the embryo than in the pericarp. Plant seeds can serve as natural vectors for vertical transmission of beneficial endophytes that confer disease resistance. Evidence showing the transmission of microorganisms from seeds to the developing seedling was found to occur in experimental and natural conditions. It also seems that the transmission to new plant happens through specific mechanisms where certain microorganisms migrate from seed to plant leaves and other to plant roots. Plant holobiont. Since the colonization of land by ancestral plant, selective pressure acting on holobiont components has likely shaped plant-associated microbial communities and selected for host-adapted microorganisms that impact plant fitness. However, the high microbial densities detected on plant tissues, together with the fast generation time of microbes and their more ancient origin compared to their host, suggest that microbe-microbe interactions are also important selective forces sculpting complex microbial assemblages in the phytosphere, rhizosphere, and plant endosphere compartments.